two different aspects of exercise to worry about, and that's that you can go from sort of rest to maximal exercise, and then that there's also training effects. So that was that. This one, pretty simple, just an algebraic description of the movement of any gas across a membrane. So this could be a membrane in the lungs, like oxygen going across the alveoli into the blood. It could be a membrane near a muscle where oxygen is going across the muscle cells or the capillaries to be used in the mitochondria. It could be anything, but real simple. There are three factors that will affect the rate of diffusion of a gas. The more area you have, the higher the rate of diffusion. The smaller the thickness, the greater the rate of diffusion, and then the greater the concentration difference across it, the greater the rate of diffusion. And that's all collectively known as Fick's law of diffusion. Pulmonary surfactant is a substance that is secreted by the type 2 alveolar cells. Pulmonary surfactant breaks up the water tension in the alveoli, which acts as sort of a vacuum trying to keep the alveoli compressed together. So having pulmonary surfactant makes it easier to inflate the lung. And so if you didn't have pulmonary surfactant, then the work of breathing is harder. And so that happens in premature newborns because they don't have pulmonary surfactant. It happens in the elderly because they don't make as much surfactant. And it just means that inflating the lung is hard work. There are a bunch of diseases that are usually characterized in ventilation. Obstructive lung diseases have to do with moving air back and forth in the tubes. Emphysema, or COPD, is chronic obstructive pulmonary disease, so you'll never forget that one. And in emphysema, it's a little bit complicated, but basically you end up having to breathe hard to exhale, and that really strong, forceful exhalation can make the tubes collapse. Asthma is pretty easy to understand if you have a muscular constriction of the tubes, because there's muscle that goes around it, then those tubes get small and it's hard to breathe. And then for edema, you simply have fluid in the way, and that makes it hard to move air back and forth. So all of these are considered obstructive lung diseases. Restrictive lung diseases are just things that make it hard to move the walls of the lungs themselves. So it's hard to inflate against kind of the stretchy rubber band of the lung. So pulmonary fibrosis makes the lungs stiff and kind of rigid so they don't inflate. And then respiratory distress syndrome, like we just said, makes it hard to inflate. Like the rubber band is a lot thicker as far as trying to stretch it. All right, for oxygen transport in the blood, it's generally not very soluble, so it doesn't go into the plasma portion very easily, so you have to have hemoglobin. Hemoglobin carries, about 96% of the oxygen in the blood, and there's a relationship between the binding onto hemoglobin and the partial pressure of oxygen, which is PO2. The hemoglobin can be anywhere from 0 to 100% saturated, and two spots to sort of recognize on here is what we consider fully oxygenated blood has a PO2 of 100, and that corresponds to about a 98 or 99% saturation hemoglobin. So, okay, that's the upper end. The lower end, which is where mixed venous blood, systemic venous blood is a PO2 of 40, and that is about 75% saturated for hemoglobin. So things to remember down here. Alveolar venous blood is coming back from the lungs, therefore it is fully oxygenated. It's going to become systemic arterial blood, and it has a PO2 of 100. Alveolar arterial blood is going out to the lungs. It's deoxygenated because it used to be systemic venous blood and has a PO2 of 40. In exercise, there's a lot of different things that lead to gender differences, and here they are. For numbers in the cardiovascular system to remember for oxygen and for CO2. So PO2 of air is 160 at sea level. By the way, on top of Everest, this is only about 50. In the lungs, it's 100, and that's because it's about halfway in between deoxygenated blood, which is 40. So 160 in the air, 40 in the blood, about 100 in the alveoli, and this is just kind of smack dab in the middle. For CO2, a little bit more complicated why, but the low end is 40, and then the high end, when it picks up CO2 from the cells, is 46, and that's kind of all you need to know for those two. All right, carbon dioxide as a gas can be dissolved in the blood pretty easily. It doesn't stay that way because it dissociates into bicarbonate, and it can also bind to hemoglobin as CO2. So here's sort of a summary of all the different binding to hemoglobin, whether it's oxygen, which can do that, or CO2, which can do that, at two spots. Near the cells where you're generally delivering oxygen and picking up CO2, and in the lungs where you are picking up oxygen and offloading CO2. Okay, so summary of all the different places. Sorry, my dogs are super annoying, so you're gonna hear them all day long. For exercise capacity, the metric for that is VO2, and VO2 max is a state that you can achieve where your body is either at its limit for delivering oxygen or at its limit for consuming oxygen, but at any rate, you hit a spot where you cannot move any more oxygen through the body as a whole. You can improve this with training, and heart rate tends to basically scale exactly with your effort. So the harder you are exercising, the higher your heart rate is, approaching your 100% max. Anaerobic metabolism is ever present. As you approach VO2 max, you get to an anaerobic wall. This can be pushed further back with endurance training. We already did this one, perfusion and diffusion limitations. This is the contribution of a variety of different stimuli to making you take a breath. The main one is this one right here. Carbon dioxide builds up in the body every second that you have metabolism functioning. And that CO2 in the blood is not great, but it can be buffered in the blood by bicarbonate. But that CO2 goes into the brain and it becomes hydrogen ion, acid, and the brain really doesn't like that. And so the central chemoreceptors in the brain stimulate the medulla. Remember, that was a part of the brainstem, which controls the muscles of ventilation, and it forces you to breathe to get rid of the CO2 so your brain is not acidic. So this is the most typical, really strong axis. Your kind of day-to-day, second-to-second control is this. There is a weak stimulus from the CO2 onto the peripheral chemoreceptors, which are in your carotid arch, and they also contribute acid, and so the chemoreceptors respond to that and increase your ventilation, but this is a pretty weak kind of backup mechanism. If you have lactic acid from exercise or ketoacidosis from diabetes, that's a pretty strong stimulus to tell you to breathe. Now, the issue is not coming from CO2, but getting rid of CO2 will alleviate it, so this is called respiratory compensation, and you'll increase ventilation to offload CO2. If you're on top of Everest, for instance, the PO2 that you're inspiring is really low. It might be 50 or less. That is below a threshold that your body starts to hyperventilate almost out of control, because now oxygen really is an issue and you have a very strong stimulus to make you breathe more. So this is very strong, but very rare under normal circumstances. This is very strong and is very common under normal circumstances, and then everything in between kind of has different occurrences. Okay, during exercise, from rest to maximal, your ventilation, so how much you kind of breathe in and out through your mouth, starts to be linear, and then at the end it gets a little bit more kicked up, and this is hyperventilation. So everything's kind of normal, and then you hyperventilate. Proof that ventilation can get oxygen into the blood is this. This is your arterial oxygen status, so totally fine. Proof that you're hyperventilating, here is your carbon dioxide, and it starts to go low because I'm blowing off lots of CO2. Proof that you are acidifying the blood, here's your hydrogen ion, and this is going up from lactate usually. And so this is alleviated by this. It contributes to the hyperventilation. There's no need to breathe this much because your oxygen is totally fine anyway, but that's what you do. Okay. No matter what exercise you do, there's always some that you buy through anaerobic metabolism. You pay it off at the end. And so this was EPOC, or excess post-exercise oxygen consumption, or oxygen. The contribution of ATP production depends on the intensity. At low intensity, it's pretty easy to burn fat to use fat as a resource. You have lots of oxygen, you have lots of blood, everything's fine. As your intensity increases, your reliance on fat goes down. Probably not to zero, that's probably not realistic, but it certainly switches to where carbohydrate is the most important thing because it's fast, it's quick, it doesn't take oxygen, and so it can give you extra on top of kind of this baseline fat metabolism in the background. We've done this before. There's lots of different reasons that contribute to muscle fatigue. Could be lactic acid buildup, but really not having ATP is probably the killer. Hydration is important because if you're not hydrated, you overheat. If you overheat, you have to send blood to the skin to cool, and that compromises your heart rate. So either your heart rate is higher than it should be, or it's the same when you're not hydrated, but you're wasting, this power is going to the skin instead of to the muscles. And detraining happens very quickly. Stroke volume goes away, therefore cardiac output Q goes away. Your heart rate actually goes up because your heart is smaller, and so it has to beat faster at the same smaller stroke volumes. We already did this one. Okay, onto the kidney. In the kidney, this whole Bowman's capsule construction is pretty important. There are these layers here that give you a molecular sieve or filtration. The capillaries are the first line of defense. There's holes that let water with most dissolved things through. Hits the basement membrane, which is negatively charged to keep proteins out, and then hits the podocytes, which are for straining things like red blood cells, white blood cells, so on and so forth. Okay. There are also capillary forces. So the Starling's forces here, hydrostatic pressure, which is blood pressure, pushes water out. Plasmas stuck in the blood that can't filter, pull water back in. And then the presence of water already in the Bowman's capsule space pushes it back in. So the glomerulus has to fight against that to have filtration, and there's a net filtration pressure of about 10 millimeters of mercury. Okay. Concept of clearance is that certain things can be filtered and then untouched, filtered and reabsorbed, filtered and completely reabsorbed, filtered and secreted, everything in between. And examples were inulin is filtered and untouched, glucose is filtered and completely reabsorbed, urea is filtered and partially reabsorbed, and then stuff that's filtered and completely secreted, kind of depends. Could be hydrogen ion, could be bicarbonate, it's kind of all over the place. All right, make sure you know the areas of the kidney: Bowman's capsule, glomerulus, proximal tubule, loop, distal tubule, collecting duct, and the whole juxtamedullary apparatus or the macula densa. Different types of nephrons. There's the short ones called the cortical nephrons. This is about 80% of the nephrons, and this does most of the filtering of the kidney. 20% of the nephrons are juxtamedullary, and the loop of Henle here creates the salt gradient for making urine concentrated as it leaves the body. Different blood vessel systems. This is the peritubular capillaries up here for reabsorption and secretion of most normal substances. This is the glomerulus for filtration, and this would be the vasa recta. Sorry, this would be the vasa recta for creating the salt gradient in the kidney. More anatomy. We already did blood vessels. Okay, the macula densa, if we're looking at it in kind of cross section here, is a sensation of the fluid in the kidney. And if the fluid is moving very, very quickly, it means that you have a lot of high blood pressure here, and your filtration at the head of the river is very strong. So that's not necessarily great, because it means that you're probably going to lose water, and you're maybe not giving enough time to absorb things back from the kidney. So you want to reduce that. So the macula densa cells taste, as it were, this water here, and if it's salty, it means things are going by too quickly. And so I need to kind of dial down the afferent arteriole. So the macula densa cells secrete a paracrine to vasoconstrict the afferent arteriole and bring everything back down. If the salt was very low in here, it would mean that the water is sitting in the kidney and I have tons of time to pull the salt out. So I don't have enough pressure here, so I need to speed it up. So this will vasodilate, and I'll have more pressure at the head of the river. The proximal tubule is probably the most important chunk of the nephron after the glomerulus. So here I have filtration, and then immediately after throwing all that good stuff out, like glucose and amino acids and water and salt, I get it back. And the proximal tubule accomplishes that. It has lots of mitochondria. It's got big chunky cells with lots of with lots of absorption. And I'm back. All right. One of the more complicated things to try and get from the kidney is regulation of blood pressure, which the kidney influences because if the kidney holds on to salt, it holds on to water. If the kidney holds on to water, then that plumps up your blood pressure. So the kidney can influence this in a variety of ways. There is a very long pathway which will improve the kidney's ability to hold on to salt, which is regulated by a hormone called aldosterone. Aldosterone comes from the adrenal cortex, which sits conveniently right on top of the kidneys. And aldosterone makes you hold on to salt, hold on to water, and therefore it can influence blood pressure. The stimulus for aldosterone is a hormone called angiotensin 2. Angiotensin 2 used to be angiotensin 1 before ACE, angiotensin converting enzyme, made it like that. Angiotensin 1 used to be angiotensinogen, which came from the liver, and an enzyme called renin, which comes from the kidney, converted that. So this whole big long pathway culminates in aldosterone working on the kidney. Why can't we just have aldosterone? I don't know. All right, so here is aldosterone and its effect. If you have more aldosterone, you reabsorb more sodium from the urine, therefore you excrete less of it, hold on to water, and it either affects your sodium balance or it affects your water balance and your blood pressure. The consequence of holding on to sodium is you have to get rid of potassium. So this is one place where it's a either or. There is no, you don't get to do both of these at the same time where I can hold sodium and hold potassium. You're trading off something right there. All right. The control of urine concentration is under the influence of ADH, which stands for antidiuretic hormone. It has the name of vasopressin, which is unfortunate, but it does. And it acts at the collecting tubule, which is the last spot the urine would see in the nephron before it goes to the urinary bladder. And so the hormone ADH improves reabsorption of water from the nephron back into the blood. It comes from the hypothalamus, and when it arrives at the tubule cells of the kidney, it promotes the insertion of these things, which are called aquaporins, which are right here. And these aquaporin molecules allow water to get pulled into blood. Now, why would water go that way? Because I have salty cells, I have salty interstitial fluid, and I have salty blood. How did I get salt here, and salt here? That's what the loop of Henle and the juxtamedullary nephrons do, is make this salty, make that salty, make this salty, and therefore water in the urine will get pulled out. Ta-da! Okay, the effect of alcohol is to promote urine formation because it interferes with ADH, so you don't get as much ADH. The effect of caffeine as a diuretic is similar, but no one understands why, and neither do I. All right, on to the reproductive system. Male anatomy, boring and easy. Male accessory glands, boring and easy. The female reproductive system, the ovaries are responsible for developing eggs and releasing one, usually, sometimes more, a month. The uterine tubules are the site of fertilization and the transport of the egg to the uterus. And the uterus should be the site of implantation, and technically the sperm passes through on the way from the cervix up to the fallopian tubes onto the ovaries. And of course, this is where you'd have development of a fertilized egg. The ovarian cycle, which regulates the release of the egg, otherwise known as ovulation, has two phases, follicular and luteal. The follicular is pre-ovulation. This is after ovulation. This is always 14 days, so ovulation begins, 14 days of luteal phase, and then you start all over again with a brand new follicular phase, which can be any number, almost anything that you can think of. So you can have a 7-day follicular phase, you could have a 25-day follicular phase. This is very random, and this is always fixed at 14 days. Here's what oral contraceptives do. We've done it a bunch of times. Here is hormones that regulate ovulation, as well as hormones that regulate ovulation and control of the endometrial lining. Here is the thickness of the endometrial lining on one menstrual cycle from 1 month to the next. Luteinizing and follicle stimulating hormones come from the anterior pituitary. They spike right before ovulation, as does estrogen. And then estrogen and progesterone carry on and keep the uterus sort of ready for a pregnancy. If there is no pregnancy, this goes away, that goes away, that goes away, and then menstruation starts and you do the whole thing over again. Fertilization involves the release of the egg, that's ovulation, picking it up by the fimbriae of the fallopian tubes, fertilization by one sperm, hopefully not two, that's bad, one sperm. You make a diploid zygote here. It moves down the ampulla right here, begins to divide, implants into the wall of the uterus, and then would develop a placenta and become a fetus. Okay, on to endocrinology, classification of hormones based on what they are. Steroids all come from cholesterol, whether it's dietary or you make it. The rest are all peptides. These are just examples of all the different ones which you should know. Here is the anterior pituitary. It is influenced by the hypothalamus, so hormones come from the hypothalamus and then act on the pituitary. So here are the hormones that influence the pituitary. They generally stimulate, but sometimes inhibit, the 6 hormones of the anterior pituitary, so FLATPG, FLATPG. And then here are other endocrine glands that are influenced by these hormones and the other hormones that they secrete. These are therefore trophic hormones because they influence the pituitary. These are trophic hormones because they influence all these different endocrine glands right here. And then these are the final non-trophic hormones because they act on non-endocrine tissues. Hypothyroidism is caused by lots of different things. The symptoms are these. Hashimoto's is the most common in the US. It's an autoimmune disorder. Cases. Graves is the most prevalent hyperthyroidism. The pancreas is involved in sugar metabolism, so glucagon and insulin sort of work against each other. This is where it comes from. These are all the different things it does. Diabetes is either because you don't have insulin made from the pancreas, that's type 1, juvenile onset, also known as insulin-dependent diabetes mellitus. And then type 2, which is adult diabetes, is resistance to insulin by the cells, and we don't know why, but triggered by poor diet and a sedentary lifestyle. The Malamute has to go poop. The 3 hallmarks of diabetes mellitus are polyuria, polydipsia, and polyphagia, having to pee, being thirsty, and being hungry. From the mouth down to the stomach, salivary amylase is working on carbohydrates, then it gets destroyed in the stomach. There's different cells that line the stomach that secrete different things. So parietal cells secrete hydrochloric acid and intrinsic factor, which allows you to absorb vitamin B12. Chief cells secrete pepsinogen, which becomes pepsin, which helps with protein metabolism. And ECL secretes histamine, and we didn't really go over that much. Mucus is an important defense mechanism, so mucus cells secrete mucus or mucin. All right, stimulation of hydrochloric acid is under hormonal influence. Histamine promotes it, so you can block that with some anti-acid things such as Tagamet and Zantac. And there is a nervous stimulation of this as well, which comes from the parasympathetic rest and digest. The importance of hydrochloric acid: the low pH helps to denature proteins so enzymes can get to them. It activates pepsinogen, which enzymatically works on proteins. Pepsin comes from the chief cells as pepsinogen. Here is the parietal cells making hydrochloric acid that makes pepsin, and then pepsin works on proteins and makes them into small peptides. All right. The dangers of having acid and pepsin, both of these are corrosive things. You defend against it with mucus. You defend against it by hopefully not letting it get to the soft tissues underneath. And then you turn over the cells frequently so that they can basically tolerate the constant abrasion. Most things are not absorbed in the stomach. Proteins are digested there. Starches are digested and then it stops. And the only things that get absorbed across the stomach are alcohol and non-steroid anti-inflammatory drugs like aspirin. And that's it. So peptic ulcers are fairly common. They are caused by an erosion of the mucosa, either by the hydrochloric acid or bacterial infection, which reduces the mucus so that the acid can get through them more easily. So you can either treat it with antibiotics or you can try and prevent the hydrochloric acid, and that's what Prilosec does. Gastritis is an inflammation of the submucosa, which is underneath those sort of more durable cells. And that's all I'm going to say about that. This is now the small intestine. The small intestine has villi, which are these folding things here, which gives you lots and lots of absorption, and microvilli, which are these, which would be on the tips right there of all of the cells that line the fingers that stick up. So villi, columnar cell, and then microvilli on the end. Liver has lots of secretory functions, including bile salts. Pancreas has lots of enzyme functions and bicarbonate, and then there's also going to be a filtration of things back to the liver by the hepatic portal system, which I didn't show on another slide. Carbohydrates, you can eat lots and lots of different types of carbs: sucrose, lactose, maltose, starch, glycogen, all generally digestible. Cellulose is not. Lactose, in people who lack the enzyme lactase, can no longer digest it. And we have to break down all these things into either glucose, fructose, or galactose. Okay. Proteins have to be broken down. Pepsin does it in the stomach, then trypsin and chymotrypsin in the small intestine. And carboxypeptidase is the enzyme that breaks down tiny free amino acids from all the giant peptide fragments that you get. Free amino acids can all be absorbed. Sometimes you can absorb 2 and 3 amino acids together, but you are generally going to break those down into individual amino acids later on. Okay. The absorption of fat requires bile salts because fat globules are big and hard to get kind of your mouth around. It's like a giant gobstopper. So making tiny little bites out of it with bile salts emulsifies the droplets into small things. Then those droplets get worked on by the enzyme lipase, which breaks off triglycerides into monoglycerides and free fatty acids. These you can absorb. Coincidentally, you're going to reassemble them into triglycerides in adipose or the blood or muscle, but you have to absorb them independently. And so here's the whole process of emulsification and absorption of fats. Vitamins, there's fat-soluble and water-soluble vitamins. There it is. Minerals and water are all absorbed in the small intestine. The pancreas secretes pancreatic juice, which is bicarbonate and digestive enzymes like trypsin and stuff. And here they are. So trypsin, carboxypeptidase, lipase, pancreatic amylase, and then the RNA and DNases. Here's trypsin activation. It starts as trypsinogen. It gets converted to trypsin by the enzyme enterokinase. Sorry, all of the dogs need to go out, and all the dogs are annoying now. The liver, I guess I did have it in here. Here's the absorptive part of it. Remember, it breaks down red blood cells, and it collects the blood coming from the digestive system before it goes back into the venous system. It kind of filters and screens that, which was here. So here is the reabsorption of blood from the small intestine, goes into the liver, and so the liver is screening out a lot of stuff and can detoxify stuff before it goes into your general venous return, which is nice. Your liver is very important. Ulcers happen when you corrode the lining of the stomach and sometimes the small intestine, so hydrochloric acid, bacteria, all those different things will do that. And ulcers are bad. If you have it puncture all the way through, that's called a perforated ulcer, serious to fatal. I think that's it. Good luck. See you guys on Thursday.